Hello everyone, so today we're going to talk about different types of mutations, so point number 3. Okay, I'm going to switch to my paint file. So, point mutations, they only are dealing with one nucleotide. So either a nucleotide has been changed, this is called a substitution, or it can be a nucleotide that's been added, it's called an insertion, or a nucleotide which has been removed, a deletion. Okay, to start off with, we're going to have a look at what happens if there's a substitution, because it can have different consequences. Okay, let's start. So there are different types of mutations. We're going to have a look at a nonsense mutation. This is where a stop codon appears. So let's find examples using the genetic code. So stop codons, we have them here. So for example, UAA is one. Okay, I'm going to write that down. UAA is one type of stop codon, or we can have UAG. Okay. So these are stop codons. So basically we only need to change the last one. So if we take UAC for example, UAC is tire, that is an amino acid. So therefore, the corresponding DNA sequences on the template DNA will be ATG, ATT, ATC. So what we're saying here is that, let's say in a DNA template you have ATG, this would give you as RNA UAC, this would give you the amino acid UAC type. So if there is a mutation, this G is changed for example into a T or a C, ATT, the corresponding RNA will be UAA, and for ATC UAG, and UAA and UAG, they're both stop codons. So if a DNA sequence has a mutation like one of these, the G replaced by a T or a C, this will mean that the RNA will have a stop codon. So depending on where this mutation occurs, it will have a more or less big impact. If the stop codon is at the beginning of the mRNA, that can have a huge impact, because that means that your polypeptide, instead of being hundreds of amino acids, it can only be 10 or 20. If the stop codon is towards the end, your polypeptide will only be missing a few amino acids, so that might not have such an impact. Okay, so that's the first one, nonsense mutations, stop codons. Okay, next one, missense mutations. So this will provoke a change of amino acid. So well, we saw an example of that, change of amino acid. That example we can simply use the one in your lesson about sickle cell anemia. Here we have a change of, we have a mutation T into A, and changing that, instead of having the glutamic acid, we have a valine. So the example in sickle cell anemia is actually, it's an example of missense mutation. So what you can do is maybe try and find another one in using the genetic code. So missense mutation, change of amino acid, that's the example of sickle cell anemia. Okay, same sense mutations. So these mutations have absolutely no impact because the amino acid will be the same. So let's take, for example, here we have val, 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 val. So to get to that, if we think in terms of RNA, we're going to have G, U, and all the 4 other letters. So G, U, for example, U, and G, U, C. So if we have this as RNA, for DNA we're going to have CAA and CAG. Okay, so here the mutation, the point mutation, is also a substitution, like all these 3 cases. So this time A is changed into G, but that will have no impact because it will be GUC instead of GUU, and this will correspond to the amino acid val. So same sense mutation doesn't change the amino acid, so therefore it will have no impact on the polypeptide and no impact on the protein. Okay, last thing, if I switch back to your lesson, frame shift mutations. So these are due to insertions or deletions, so when a nucleotide is added or deleted. This can have quite a big impact. We're going to see why. So let's take this sequence of RNA which has occurred after normal translation. So we've got ACG, AGG, ACU, etc. So T with the corresponding amino acids, THR, ARG. So the first case we're going to have a look at, imagine that this nucleotide here, adenine, is deleted. It's deleted from the sequence. This can be due to the fact that sometimes DNA polymerase skips one. We did say that errors do occur, so sometimes a nucleotide can be skipped. Therefore, in the RNA you'll have a missing nucleotide, and therefore where the starting point is going to shift to another nucleotide, starting on the C this time. So instead of reading ACG, as the A is not present anymore, we're going to read the ribosomes are going to read CGA, and therefore we're going to have ARG, then we're going to have GGA, GLI, etc. And if we compare these 2 sequences, we've got completely different amino acids. Here we have THR, ARG, THR, ALA. Here we have ARG, GLI, LU, etc. So these 2 polypeptides will be completely different. So that means that they will, if they're enzymes, for example, they're not going to do at all their function because their active site will be completely different. So different polypeptides, different 3-dimensional shapes. So frame shift mutations, they're going to change completely the protein, and basically the protein will either be very differently functional or non-functional at all. Again, these frame shift mutations, a bit like the nonsense mutations, it depends where they occur. If they occur at the beginning of the mRNA, then the consequences will be dreadful. That will completely change the polypeptide. If they occur towards the end, if only a few amino acids are changed, that might not have so much of a consequence. Second example is just A and C that have been deleted, and it's again completely different. Please note, if we take 3 out, so these 3 for example, A, C and G, that's only going to take 1 amino acid out. So you will still have ARG, THR, so sometimes that will not have so much of an impact. So there you go, those were the different types of mutations. Goodbye.